Amir is the director of the Brain Imaging Signals Lab. He's a professor at McGill University in the Department of Neurology, Neurosurgery, Physiology, and Biomedical Engineering. And he started his research career at the Hebrew University in Jerusalem before completing his PhD at the Weizmann Institute. During his postdoc, he investigated neurovascular coupling and the mechanisms underlying high-resolution fMRI at CMRR at the University of Minnesota, as well as at Max Planck Institute for Biological Cybernetics in Tübingen. His current research focuses on understanding the functional brain imaging signals that we all record and their relationship to underlying neuronal activity. To do so, he employs layer-specific neurophysiology, neuroimaging, and computational modeling. Today, he's going to talk to us about the complexity of lamina-resolved neuronal activity and the spatial specificity of bold, CBV, arterial, and venule responses, as well as the implications for planning and interpreting layer-specific studies. So Amir, you are
Tyler.
Perfect.
Tyler, can you hear me? Can you hear me?
Yep, we can hear you, and
Great.
We are all ready to go.
Right. So you presented the complex title, and I'll move immediately to the simpler title, what we can and we cannot do with the presented fMRI. So of course, when we introduce subject in the scanner, we present stimuli or task, we get bold signals or CBV signals. In between, there is a process, a very complex process of neurovascular coupling. Many laboratories investigate that, and then MRI, PET, or optical imaging to do the physical measurement. Most of us are interested actually in neuronal activity. We use functional MRI because it has the best spatial specificity and spatial resolution relative to all non-invasive imaging methods. So this transformation is extremely important. Overview. So that's the long overview, but really I can split it according to colors to talking about a Lamina-resolved, like general, the methods that we use in the lab. Low-pass filtering and undersampling in space, low-pass filtering and undersampling in time. Some of these items are going to be really simple, but I'm not sure that we all think about it. Neuronal activity reflected in fMRI responses. Cortical layers are densely interconnected. What can we do? So here are the methods that, the neurophysiology methods that we use in the lab. So we use mice, rats, and recently Laminar specific also in monkeys after an unbelievable delay in getting infrastructure for monkeys. We use laminar probes, as you can see here. So from this point to this point, they are 3.1 millimeters, and the contacts are separate, adjacent contacts are separated at intervals of 100 microns. We are approaching even a higher sampling rate. Here is an example from a rat where we see the cortical vasculature, the pial vasculature in dark curves, and the gray area is actually cortex after removing the skull, and in the case of rats and monkeys, also the dura mater. And here's our probe, and we can see 4 contacts, the upper 4 contacts above cortex, and there are 28 below or inside cortex and below cortex. So we sample all cortical layers. We also do our best, and we actually succeed, to insert probes very precisely, in this case, to one single barrel, a predetermined barrel in a mouse, which is ellipse of 200 by 300 microns. Then an important part of how we study laminar-specific studies is that in acute and also chronic experiments, we perform lesions by micro-injection or injection of microcurrent at the end of the session. So here you can see 3 lesions in rat somatosensory S1FL. And with these lesions, at least these lesions allow us to solve some equations, linear equations, in order to localize the probes at a very high precision. So the red contacts are those that we use to inject the microcurrent from, and then by interpolation, extrapolation, and optimization, we can really localize all the other contacts very precisely in cortex. So first, about Lamina-resolved spatiotemporal neurophysiological responses. So, just a reminder, with every small voxel, even if it is 0.5 or 0.5 or 0.5, we sample a huge number of neurons and synapses. Here's what we get with neurophysiology. This would be the multi-unit activity response to 4-pulse stimulation of the rat, very similar to median nerve stimulation in humans. That would be the multi-unit activity in the middle, local field potential, and then current source density, which is a model that approximates the synaptic currents that go into and out of the population of cells at each location. As you can see, at least in this case, we cannot learn much from multi-unit activity. Also, local field potential is not giving us a lot of information on layers. And in fact, we measure local field potential. The spatial specificity of local field potential is very poor. And the reason is that the cortex is a volume conductor. So any contact in cortex can detect the local field potential of a source, which is reasonably far away, at least 1 millimeter or even more than that. On the other hand, current source density not only give us a very good biophysical measure of neuronal responses, it's also very spatially specific. Here is after a lot of averaging, the current source density from a rat area S1FL in response to 4-pole stimulation. Can fMRI approximate Lamina-resolved? Can fMRI approximate Lamina-resolved neuronal response in space? So the distance in S1FL from the surface of cortex to the gray matter, white matter, is about 2.1 millimeter. By the way, you can see here in the dark dashed curves, the layers that we obtain with the lesions and modeling, et cetera, et cetera. You can see how complex the current source density response is. Blues are a depolarization, excitation. Reds are a hyperpolarization. And you can see how complex it is. And now if we superimpose here, now I cannot really move. That's the zoom things. Okay, yeah. If I just throw in 4 voxels that are 0.6 millimeter isotropic, that's what we get. Obviously, low pass or we undersample, and we have here a large partial volume effect. Now, specificity of cerebral blood volume and bold responses. This is a study done in the cat by Zeshan Yao, a PhD student in my lab, and myself, where we image cat area 18. We look here again at the pial blood vessels. We segment the vessels, classify them according to arteries and veins, and we can also measure the diameter of the veins and look at the classes starting from really very small all the way to relatively large diameters in veins and arteries. We presented to the cat gratings, and we computed differential orientation maps as band match to delineation of ROI first, but also in order to estimate the selectivity of gray matter and blood vessels. As you can see here, selectivity, we define selectivity as the response, the fractional response to the optimal orientation and minus to the non-optimal orientation. That would be the result with HBr, deoxyhemoglobin that is an approximation to bold, and this would be the response with HBT. So first, I want to demonstrate the result of HBR. We demonstrate here the response in blood vessels, on top in veins and at the bottom in arteries, relative to 1, which in this case is the spatial specificity in the gray matter. So we see that we get responses, selective responses that are also there, even with relatively large venules of 0.2 or even 0.3 millimeter diameter. We also get similar phenomenon, although to a lesser extent, when we look at cerebral blood volume with HBT. We do get selective responses from veins and arteries, but even for reasonably large arterioles. The important thing that I want to mention here is that if we now compare directly the spatial selectivity of HBR, both relative to blood volume, we see that in gray matter, we get about 0.55 in vein. In gray matter. That's what I want to emphasize here, gray matter, which means that the spatial specificity of a cerebral blood volume is much higher than that of bold responses. We also see the ratio in arteries and veins. Both arteries and veins do, as I said, mention or show selective responses. But the main point here is that looking at very high resolution and very high signal to noise ratio in CAT area 18, cerebral blood volume responses are much more selective than bold responses. Can fMRI approximate Lamina-resolved neuronal responses in time? What we see here are stimulation of whisker in the mouse. That's a single whisker stimulated with 32 channels inserted to the brain. All of them in the mouse, all of them sample cortex, because the interval between contacts is only 50 microns. And we stimulate at a rate of 2 hertz. We see that the responses are very swift. They are really swift. At the bottom, you can see a magnification of only 100 milliseconds with signals sampled from specific layers, and more or less at 35 milliseconds, the response is gone. And of course, there is information flow between layers. Needless to say, we cannot reproduce that with a functional MRI. Transient and steady state responses. So that's a comment that I want to mention about the type of stimuli that we use. If we use transient stimuli, such as deflection of a whisker, like we present here, we get a transient response, which means that in between the transient stimulation, all we get is actually bold response to spontaneous activity, not really what we want if we are interested in the response. On the other hand, if we use, so these are transient responses. On the other hand, if we use a stimuli that produce a steady state responses, like these stimuli here, which are rotating checkers, then we get over 20 seconds, we get steady state responses. If we have lamina-specific responses here, we will be able to get the lamina-specific response for this entire time. This is local field potential, this is multi-unit activity. This was done with a single electrode. Complexity in time, Laminar-specific phase-amplitude coupling during spontaneous activity. So if we want to look at spontaneous activity with a high-resolution fMRI between layers, there are more complex issues when we look at the neurophysiological signal. So for example, if all of these are 32 channels in rat area S1SL, and at the bottom, we show the current source density in the alpha band after we aligned layer by layer or contact by contact according to the transition between sync and sources. Okay, so everything here is aligned. And then we plot the power, the amplitude of current source density in the fast gamma, middle gamma, low gamma, and beta amplitude. You can see that the phase of alpha modulates the power, the amplitude of higher frequencies. And that's true not only for alpha, it's also true for delta and for theta. So that adds to a complexity which fMRI is transparent to. Then, local field potential, current source density, or multi-unit activity, which neuronal activity is reflected in fMRI response. I would say we are still not confident about that. Logothetis et al. 2001 is supported very much that local field potential of the bold response reflects local field potential. However, his results were not really very strong. And later on, Shmuel and Leopold showed that during spontaneous activity, the response in gamma local field potential and in spike rate are correlated equally to bold fluctuations of spontaneous activity. So we don't really know, not only in general functional MRI response, but also in laminar specific functional MRI response, which makes the modeling even more complicated. Last, cortical layers are densely interconnected. Here you can see a scheme of connections that were detected both by previous anatomical connections and slice stimulation connection in vitro, slice stimulation in vitro, between excitator neurons on the left and between inhibitory neurons on the right. And obviously there are 8 different populations here, and also the thalamus, and all of them are interconnected. The thickness of the arrows show the relative strength. So, you know, once we stimulate all these connections immediately within milliseconds, the entire cortex is active. And we don't know excitatory or inhibitory, etc. That's very complicated. Interestingly, John presented just before me his proposal for biophysical models, and I agree this is a very powerful tool. Sotero et al., Sotero from my lab and colleagues were able to reconstruct these connections simply by using a biophysical model whose input is the current source density response in different contacts. So I agree, this is a very powerful approach, but of course, we still have a long way to go. So conclusions or preliminary conclusion, can fMRI approximate Lamina-resolved neuronal responses in space? We undersampled the response. By the way, this talk was motivated by a discussion in the previous dinner, in which there was a discussion whether 3 voxels are possibly sufficient. So I commented in the discussion, no. And to myself, I asked, what's going on with Renzo? Does he want to retire? He just started. So in any case, can fMRI approximate a Lamina-resolved neuronal response in space? We undersampled the response, spatial specificity. Not only we undersampled, I didn't talk much about blood vessels, but obviously, given what I and other people showed about blood vessels need to be masked out, either by SWI or a different method, veins, venules, and if we look at CBV, also arteries and arterioles need to be masked out. Spatial specificity. In our hands, CBV is more specific, and as I said, we need to mask out vessels. Can fMRI approximate Lamina-resolved neuronal responses in time? That's of course difficult, but if we use steady state responses, stimuli that cause steady state responses, we have better chances of resolving the lamina in space, and not necessarily in time, because in time, in any case, it's steady state. local field potential, current source density or multi-unit activity, neuronal activity reflected in layer-specific fMRI. What is it? That adds to the complexity. Cortical layers are densely interconnected, and this functional connection, or functionally, these connections are reflected immediately. So I'm recruiting too. You can see here the different projects, and I want to acknowledge CIHR, NSERC, my funding agency, and I would like to thank you for your attention.
Thank you very much, Amir. That was really, really nice. Are there any burning questions from viewers while we set up for the next talk? If not, I have a really quick question, which is, it actually would have applied to Jonathan's talk as well, but as you start looking at the really extreme details of some of these responses, do you feel like there's kind of a sweet spot for the generalization that you're trying to achieve in order to make it like generalizable to many, many applications? We talked today a lot about detailed responses, and these are really interesting for our field as layer-fMRI investigators. But of course, fMRI is used for a lot of different things. So Amir and Jonathan, if you have any comments.
So to me, your question is unclear. You ask about generalization, but generalization with respect to what? I presented some 7 different items very quickly. So generalization with regard to what?
No, that's a good response. I mean, obviously, you also have to balance all the different items that you're talking about and which one you want to try to push. For this is the most important to have the detail that still generalizes, while others might fall wayside, of course.
I would like to mention that at the end, we need to remember that until the gradient system, which is being developed in Berkeley and at Mass General, appears, we sample about 3 voxels in cortical layers, and they represent really a very complex underlying mechanism. So we get some reflection, and maybe we can, diagnose neurological conditions with them, just like is being done with other methods in structural and functional MRI. But they are far from really reflect the mechanism in a very precise manner. They reflect something. What exactly, we don't know yet, in my view. And even when we get this gradient system, so I think it's going to be at best 5 voxels, but it doesn't change much the picture.
Thank you. Natalia has raised her hand. And in the meantime, do you mind taking down your slides so we can set up for the next talk? Perfect.
Hi, Amir. Thank you. Very nice talk. I was wondering what your thoughts are about glial cells, or astrocytes, actually, specifically. So we know that there is a metabolic contribution in your neurovascular coupling specifically, and probably blood flow.